Cell cultures are utilized in a wide variety of research and diagnostic applications. It is sometimes desirable to store cell lines for future work. To preserve cells, avoid senescence, reduce the risk of contamination, and minimize the effects of genetic drift, cell lines may be frozen for long-term storage. In this video, we show you how to freeze cells grown both in monolayer and in suspension, and how to thaw them for growth once again.
Hi, I'm Ricky, here at Dr. Phelan's laboratory at Molecular Pathology Laboratories Network in East Tennessee. Today, I'm going to show you how to freeze cells for long-term storage and how to thaw them from liquid nitrogen.
To freeze human adherent cells, you start by lifting them from the plate as you would if you were passaging them. Keep in mind that it's best to use cells that are in their log phase of growth for cryopreservation. Once the cells have detached and you've stopped the trypsin reaction by adding serum or medium containing serum, transfer the cell suspension to a sterile centrifuge tube and add 2 mL complete medium with serum. Cells from 3 or more dishes from the same subculture of the same source can be combined into one tube. Centrifuge the cells for 5 minutes at 300 to 350 Gs at room temperature. After the spin, remove the supernatant and resuspend the pellet in 10 mL of 4 degrees Celsius complete medium. Add an equal volume of 4 degrees freezing medium. Mix the cells thoroughly and then place the tube on ice. Count cells using a hemocytometer. Dilute the suspension with more freezing medium in order to get a final cell concentration of 1 million or 10 million cells per milliliter. To freeze cells from a nearly confluent 25 centimeter square flask, resuspend in approximately 3 mL of freezing medium. Pipette 1 ml aliquots of cell suspension into labeled 2 ml cryovials. Be sure to tighten the caps on the vials. Place vials 1 hour to overnight in a -80 freezer. Then transfer them to a liquid nitrogen storage freezer. Freezing cells from suspension culture is similar in principle to freezing cells from a monolayer. The major difference is that suspension cultures do not need to be trypsinized. First, transfer the cell suspension to a centrifuge tube and spin for 10 minutes at 300 to 350 Gs at room temperature. Next, remove the supernatant and resuspend the pellet in 4 degrees freezing medium at a density of 1 million to 10 million cells per mil. Then, transfer 1 ml aliquots of cell suspension into cryovials and freeze as done for the monolayer cultures, which was demonstrated in the previous chapter. To thaw and recover frozen vials, first remove the vial of cells from the liquid nitrogen freezer and immediately place it into a 37-degree water bath. Agitate the vial continuously until the medium is thawed, which generally takes less than 60 seconds. Once thawed, wipe the top of the vial with 70% ethanol before opening. Then, transfer the thawed cell suspension into a sterile centrifuge tube containing 2 mL of warm complete medium and 20% fetal bovine serum. Centrifuge this for 10 minutes at 150 to 200 Gs at room temperature. This step will remove any residual DMSO. After centrifugation, discard the supernatant and gently resuspend the cell pellet in a small amount, around 1 ml, of complete medium and 20% FBS. Transfer this to a properly labeled culture plate containing the appropriate amount of medium. Generally, 1 ml of cell suspension is needed in 5 to 20 ml of medium. Now you can put the cells in an incubator at 37 degrees Celsius to grow. After 24 hours, you will want to make sure the cells have attached to the plate. After 5 to 7 days, or when the pH indicator in the medium changes color, you will want to change the medium. You will want to keep the cultures in a medium containing 20% FBS until the cell line is reestablished. If the recovery rate is extremely low, only a subpopulation of the original culture may be growing. Be especially careful of this when working with cell lines known to be mosaic. If cultures need to be shipped, this can be done easily for both monolayer and suspension cultures in 25 centimeter tissue culture flasks. Cells are grown to near confluency in a monolayer or to desired density in suspension. For monolayer cultures, the medium is first removed and then the flask is filled with fresh medium. It is essential that the flasks be completely filled with medium to prevent cells from drying out if the flasks are inverted during transport. For suspension cultures, fresh medium is added to fill the flask. The cap is tightened and secured in place using parafilm. The flask is sealed in a leak-proof plastic bag or other leak-proof container designed to prevent spillage in the event that the flask should become damaged. The primary container is then placed in a secondary insulated container to protect it from extreme temperatures during transport. A biohazard label is affixed to the outside of the package. Generally, cultures are transported by same-night or overnight couriers. We've just shown you how to freeze and thaw human cell cultures for both adherent and suspension cells.
So that's it. Thanks for watching, and good luck with all your experiments.